When Escherichia coli moves in a medium that lacks a concentration gradient, the cell travel stops or tumbles and then continues moving along or running in a new random direction. When the medium contains a concentration gradient, the cell moves toward chemical attractants or away from chemical repellents. This process is known as chemotaxis.
When the cell exists in an environment containing attractants, the cell will move toward the higher concentration of the molecules. This is known as positive chemotaxis.
When the cell exists in an environment containing repellents, the cell will move away from the repellent molecules. This is referred to as negative chemotaxis.
Chemicals bind to chemical receptors called methyl-accepting chemotaxis proteins, also known as MCPs, located in the plasma membrane of the cell. The cytoplasmic side of the MCP interacts with two proteins, key W and key A. Key W is an adapter protein mediating communication between receptors and key A.
When the MCP is not bound to an attractant, it stimulates key A to phosphorylate itself using ATP, producing ADP. Key A can then donate its phosphate to the response protein, key Y. When key Y is phosphorylated by key A, it moves to the flagellum. This causes the flagellum to rotate clockwise and tumble. Thus, a decrease in attractant molecules promotes tumbling.
The phosphate on key Y is then removed by key Z. This action stimulates the cell to run.
With no attractant bound, key Z is constantly dephosphorylating key Y, producing a normal run-tumble swimming pattern.
When an attractant, labeled ligand A, binds to the methyl-accepting chemotaxis protein, key A is unable to phosphorylate itself and therefore is unable to donate its phosphate to key Y. The lack of phosphorylated key Ys results in counterclockwise rotation of the flagellum and the cell runs. This allows the cell to move towards the higher concentration of the attractant molecules.
The protein key R adds methyl groups to the cytoplasmic portion of the MCP. When the attractant ligand is bound to the methylated MCP, key A reassociates with the MCP. Key A phosphorylates itself using ATP and then donates the phosphate to key Y. Key Y moves towards the flagellum. Once bound, phosphorylated key Y promotes tumbling.
The protein key B is phosphorylated by key A. The phosphorylated key B removes methyl groups from the methylated MCP. Key A then dissociates from the MCP and can no longer phosphorylate key Y. Therefore, the cell runs.
When a repellent molecule labeled ligand R binds to the MCP, key A is able to phosphorylate itself using ATP. Key A then transfers the phosphate to key Y, which moves towards the flagellum and promotes tumble. This allows the cell to move away from the high concentration of the repellent molecules.